Okay, so welcome everyone to this ReDU workshop, or this just workshop general and series. Um, so I won't steal too much of Alan's thunder when he introduces it, but broadly, prior to this, we showed you some of the analysis tools and how you can take advantage of some of the public data. But one of the key things that's missing is how do we know anything about the public data? And also how can you empower sort of this reanalysis and comparing more thoroughly your own data to what's already out there. And there's like 1,500 data sets, couple hundred thousand, probably 200,000 actual LCMS runs. So it's a lot. Not big compared to genomics, but you know, we're getting there. And so Alan, Julia, and Christy, they'll give you a little primer on how to use it. And hopefully you'll be convinced on why you should use it, or at least a little bit of that. So thank you all for coming, and let's get the show on the road.
Awesome, thanks. And thank you all for attending. I know for some of you it's both early and late. And kind of what Ming said, there's a lot of public data that's already available in mass spec, well, particularly in metabolomics, and that's ever growing. But we really need ways of really capturing and utilizing that information in systematic ways to empower your own research at the present. And that's kind of where the motivation behind developing ReDU basically started. And just to kind of orient people, you know, this is kind of the typical workflow of a mass spec experiment, right? Metabolomics experiment. You have your sample, you collect your data, you have some kind of output, whether or not it's a table or figure, and then if you don't deposit that data, it essentially dies, which is an incredible waste of, you know, the resources, time, and expertise that went into collecting that data. So what we want to do is make sure that we capture as much of that as we can to reutilize that in the future. Because all of you are somewhat familiar with with the GNPS platform. I'm just gonna briefly mention, you know, this gnps.ucsd.edu. And really, we're viewing this from the perspective of ReDU as the data analysis place. So this is where a lot of the nitty gritty and the data analysis functions are going to occur in GNPS. But the problem is it's somewhat disconjointed from the data repositories where the data actually lives. So GNPS and Massive are, you know, Massive is essentially the database that connects directly to GNPS. However, even though it's directly connected, it's still not trivial to find individual files within that data repository that you may be interested in reanalyzing. So for example, if you wanted to select just urine samples, currently what you have to do is browse all the different massive data sets and find the individual files that are actually urine, knowing that in a single study there's probably blanks, controls, and all kinds of other sets of files. So essentially ReDU is your shortcut in doing this. It is the mechanism by which to find that, you know, those urine files within the entire repository, as well as encompassing all the tools that we need to actually capture the metadata so that we know that that file is urine in a consistent control. We built some tools that are really unique to this kind of story scale analysis that this enables. That we'll be reviewing a little bit today, and just some bullet points. You know, it kind of follows the ethos of GNPS in terms of this is community driven and it's data driven, right? It's as powerful as the community can make it, and it's again a system for metadata collection, and there's some high level tools, and really what's I think is most interesting to people is you can reanalyze or co-analyze your own data with this. So starting to build out molecular networks using your data with the public data in a easy way, so that you can gain more information about your own system. And we have a preprint online. There is a manuscript currently under review that we hope to be able to share soon. And just to orient people, so the website is redu.ucsd.edu. Sorry, I forgot to put it on the slide, but it's located right here if you can see my arrow cursor. But on the home page, on the banner, essentially we have all the basic functions that you may want. We have documentation links. We have a specific link to the subset of documentation on how to actually add your own data to the system. So as soon as you add your data, it then becomes available for the file selector, which is used in all the re and co-analysis tools. It also becomes available in the chemical explorer, which you can use to basically find where a molecule occurs in the public data. We also have a validator. So a critical part of this platform or this workflow is really the ability to capture metadata consistently. So we have a template that you fill out, and then you can drag and drop that template into this validator, and it will basically tell you what is adherent to our recommended terminology or what is not adherent. And if you need any additional terms in order to capture your information, then there's information on how to contact us and add that. And then again, kind of following the general ethos of GNPS, everything that we have is basically there for you to download. Warning, it's large, but you can download all the metadata, as well as you can download all the library hits from all the files in ReDU as a tall table. So you can start doing your own analyses if you really want to. Okay, and this is a teaser and kind of show you what the capabilities are right now currently in ReDU. If you navigate to this button, Explore Multivariate Analysis of Public Data, it leads you to an Emperor plot. So Emperor is a visualization tool. Here we're performing PCA, which is a multivariate statistical analysis, and we're performing that on all the annotations in every file. So every dot here is a file that's available in ReDU, and then we can color all that information based on the metadata that we've captured in the systematic way. So in this case, we're looking at by year. You can look at instrument, by sample type, by extraction method, so on and so forth. And the nice thing about this Emperor plot, if you go to the website, it is interactive, so you can rotate it, spin it, and Chrissy will show you a little bit how to actually project your own data onto this reference map, if you would. Okay. Are there any quick questions before I want to jump right into showing everyone how to actually create some of this metadata? Okay. I think I unmuted everyone, or I gave you the ability to talk. Do you have any quick questions?
So maybe I want to illustrate how you might use this infrastructure. So for a metabolomics person, let's say you want to look at all public data of women under the age of 2. You know, if you were to ask that question now, what you'd have to do is download all the data sets, look at their metadata, and then see which files are of women under the age of 2. What ReDU allows you to do is basically filter based on those, and all those data sets come together. The same thing can be done for actinomycetes. If you are interested in studying actinomycetes, you can do that. If you want to study based on longitude, latitude, where samples came from, you can do that as well. And so it allows you a very efficient filter based on metadata. And so of course, the way that is done is by creating controlled metadata vocabularies that you then upload with the data set in the public domain in GNPS. And so Alan is going to highlight exactly how to create that systematic controlled vocabulary metadata template.
Perfect. Thank you. Thank you, Jacob.
Does anyone have questions? Great, then you can proceed.
Okay. Okay. Let me just. Okay. Perfect. Let me share my screen. So, the first thing. So, how do you find the template? So, if we're at redu.ucsd.edu. Sorry, I should have the URL on there. If anyone's having trouble, we have people in the chat ready to help. So if someone could post the URL in the chat to everyone, that'd be great. Actually, I can do it right now. Okay, awesome. So if you go to the ReDU, you can click on the How to Contribute tab at the top. That will link you to the documentation. And then at the very top, there's the section called Required Items and Summary of Steps. And the first thing is you need mass spectrometry data. This should be ideally converted to MZXML or MZML and uploaded to Massive. And the critical thing is we're only operating on public data. So we encourage people to make things public always. And then the way you actually get to the metadata template is through this link. So if you were to click on that link right there, it would take you to the sample information template. Okay, perfect. So that looks something like this. So it's a Google Doc. Again, if anyone's has any questions, raise your hand in chat. I think people have the ability to unmute themselves. But essentially the first step, so this is a read-only copy, this base template, but you can make your own by going to File and then going to Save Copy As. And that should create a private copy that then you can actually fill out. Okay. It's hard doing these workshops when you have no visual feedback, as Ming has said previously. Don't know where people are. So if I'm going too slow or too fast, please let me know. So once you have a copy, you can essentially start filling out all the information in the sheet. And we have, oh, sorry. We start, the basic information is essentially listed here. So we don't want to overwhelm people with a bunch of questions. We're really trying to capture the base information of and really the high level questions that people are going to ask. So the essential things that you are required are the massive ID in the first column. So if you don't have data today already in massive to add this to, then you can fill out the rest of it and just leave that column blank until you have that massive ID created, and then you can just copy and paste that into this document. But essentially the file name here is the most critical thing. That's what links all this metadata together. And it must have the MZXML extension. So as you can see in column B here, it's example file name. And then for every row, which is every sample, you can fill out all this metadata that we're capturing. And everything is a drop down. So it has specific terms that we're either pulling from an existing ontology. So for example, if it's we use the disease ontology, if you're operating on, say, clinical samples, but we also use Uberon, we use NCBI taxonomy to capture that information in a systematic fashion. And then in cases where ontologies don't exist, we actually have created our own controlled vocabulary. And that's where if you find yourself needing a term that isn't available, you can contact us and we'll add it to the template so that it's available to you and everyone else. This is a way to avoid essentially bloat and make make sure that everything is functional within ReDU for people. So if anyone has any questions, we could take a couple minutes, and if you want to just play around with it and ask us any questions about any of the meanings of the columns or anything else like that, I think that's this would be a great time to do that. And I can actually share the template in real time. Can everyone see the template?
Yes.
Okay, okay. So again, just to orient everyone.
Do you have the link for the template that one can copy? Maybe you could put that link into the chat.
Yes. That's a good idea. Let me, don't think this will expose anything. Okay. Oh, perfect. Someone actually already did it. Great. Thank you. Someone has a little bit of background. But if we're in the template, the first tab is essentially there to give you an example of what the column is intending to capture, whether or not it's required, the type of information that it is, and really, like, if it's linking to any ontology, then we provide the link for you, so that you can look up and email us. You know, if there's a term that we're not including, you can email us that term explicitly. That makes the turnaround time a lot faster. But essentially on the second tab, that's where you can input the data. So again, it looks something like this, where you can put in your massive ID, you can name your example file, and in this case, oh, this is quite slow on Zoom, I apologize. But let's say it's a blank. We'll call it a extraction blank. So there's ways of capturing not only the actual samples, but all the other surrounding technical things. And I would highly encourage people to add those as well, just so that we can, as a community, understand really what a blank means in terms of the annotations and quality, as well as it will help us attribute certain chemicals to being background. I think there's a, okay. I see. Oh, great. I see a question for run order. That's a great question. Run order, there is not currently a specific column for run order. However, I forgot to mention, and this is a great time to bring this up, you can add any additional column to this sheet that you desire. It won't be made available to everyone, but it's available for you in your own use. So if you have another subsequent question, and we have a couple of downstream tools that you could use that information in. But run order is not something that we ask broadly for everyone to input, but that is something that you could add as a separate column to try to capture that data. And we had a question about the massive ID. And yeah, the massive ID is created when you contribute that data. And when that job finishes, you will get that massive extension, and there you can plug it in right there. And Julia just shared the link to massive. Thank you. Yeah, so the follow-up question on the run order and the blanks, yeah, that's exactly why you would do that internally to your own system, you know, to understand, you know, what's a sample before and after, the blank before and after a sample. In that case, it's a little bit hard to capture that systematically through you know, an entire repository, and we didn't want to necessarily overburden people with adding the order for everything. But definitely encourage you to do it for your own sample set, if that makes sense. But again, you can just come down here and we're capturing the basic information. 2020, the sample collection method is really say how the sample is drawn. So if it's blood, you would say like it's a venous draw, right? As opposed to a capillary draw, because that may actually change some of the chemistry that's observed. We have some sample extraction conditions. They're not comprehensive, but this is one thing that commonly people ask us to add. We're happy to do so. We're just trying to make sure that we capture that information as systematically as possible. Instrument types as well. So we use the mass spectrometry ontology. So the mass spectrometers actually have a controlled ontology as well. So we're making sure that we capture that information, make sure that we see any systematic differences. We allow any type of information. So there's even GCMS information in here. So we take LCMS, GCMS, reverse phase, normal phase, direct injection, capillary electrophoresis, whatever you have coupled to your mass spectrometer, we will take essentially. And then there's some basic information as well. And just to note, these gray columns at the end, these are automatically calculated for you, so you don't have to fill those out. Those are just things that we make sure that everything is in alignment with a couple of the terms that we're using to control things. Okay. And just two further things to point out to people. There's a couple of these locked tabs. And the first one, it's just a list of all the possible terms that we're allowing per category, and these align with the sample information sheet. So here you can see basically easily all the terms that are available, and as well as yeah. So, and if you need anything else, you can request it from there. So if nobody has any questions, I'd like to show you how to upload it. We can also take a couple of minutes and give people time to play around with it. Okay. Any questions? Can you leave some columns empty? Oh, that's a good question. Yes, you can. The recommended way of inputting empty information, and I can switch back to the template real quick. There are options in every column that say something like not collected or not applicable or not specified. So not applicable, we're recommending where it's really does not pertain to your data set at all. So if you know, for example, if you NCBI taxonomy, if you're working on a chemical standard in that row, you know, it's just simply not an applicable term, right? Whereas if you're working in natural products and you've isolated a new organism, you know, bacteria or fungi, and you simply don't know what it is, you can use the not specified term. Not specified or not collected is equal. So I would recommend in that case not specified. And in cases where you may not know some of the technical details of the data, you can say not collected. That's kind of the way that I would use those. But in general, I don't think you can leave any column empty. However, when we get to the validator screen, I can show you essentially what would happen if you did leave something empty.
And each row can be different type of sample information as long as it's included within the same massive ID. That's
correct. Yeah, so we would recommend creating one of these templates per massive ID, as this template lives within each massive ID's folder system. But yes, you can have you can basically put all the files that are in that massive ID in here, even if they're discordant in terms of even time or date or run order. They just need to be, yeah, line by line, file by file. Okay. Any other quick questions? Okay, so blank QC is, yes, you can use that as a pooled QC. People have also started adding like retention time standards as that. I think that's equally okay. Capturing the information systematically about what blanks are what, we found to be very difficult, and terminology is kind of mixed and things. So that's, yeah, I think blank QC is really intended to capture the pooled QC standards, whereas some of the other standards are wouldn't fall into that, if that addresses that one question. And then Ricardo had a question. Yes, one massive ID. One massive ID can cover an entire batch of samples, essentially. You can also add a single sample to massive. That's perfectly fine too, if that's all you have. You know, we'll take whatever data in the public domain as possible. But yeah, you can add, you know, as many files as you'd like to a single massive ID. Okay. Any other quick questions? These are great. This is kind of the workshopping time for you all to get familiar with the metadata sheet and capture. So this is kind of the first step. It's maybe not the most exciting step. We'll show you the data analysis stuff in a bit here. Okay. Okay, I'm quickly move on here. And I'm gonna purposely, wait, which screen am I sharing, Julia? I'm lost.
You're sharing the website right now.
Okay, template.
Yes.
Okay, okay, cool. So here, once you've completed things, oh, you know what? I should make a copy so I don't overwrite the template. I have write permission, that would be bad. But I'm going to intentionally make an error so I can show you all what happens when you validate it. Sorry, internet is slow. Still slow, still slow. Okay, but I think this will err. We're just going to leave one blank. So when you're done with the template, we recommend deleting all the extra rows in the template. Sorry, my internet is incredibly slow right now for some reason. It's probably because my toddler's in there watching cartoons / doing preschool activities. Okay, well, we're going to come back to that in a second. Share, go back to the slides. Okay, so once you have everything filled out, like I said, we recommend that you go ahead and delete all the extra rows. And this just makes the file size smaller, as well as we've seen some issues with it. In some cases, when you download it, depending on the system, it adds nans, which basically results in a whole bunch of errors in terms of the validation. It doesn't affect the performance, it just kind of makes things ugly, and it makes things hard to assess if there are any things that you need to maybe update. But essentially, you can come in, delete the extra rows here, and then you want to go File, Download As, and then it's important that you say tab separated values. So we're operating on a TSV file format here. So exactly how the GIF displays. And these slides are available for everyone afterwards. as well as all these are on the documentation for you. Okay, so now we can go to the validator. So if you go back to the ReDU homepage, essentially all you need to do is drag and drop that TSV file into that blue box. So let's see if I can get this operating. Again, any questions throughout this, please feel free to bring them up. Well, my internet's too slow to do the download. But if you were to, so if you go to the validator, if you drag and drop in that bottom panel where it says sample information validation errors, first 500, it will display the information. The header is basically the column, the line number is the row, and then it gives you the error string. So it tells you essentially if you were to accidentally put plant or capital P for plant into the term, it shouldn't pass the validator and the Google Sheet. It'll give you a warning. But let's say if you get all the way through and that doesn't occur, if you do the validation, it will tell you, okay, in the sample type in row 3, you accidentally put capital P for plant instead of lowercase p. In that case, you can go back through, change that in the sheet, and then revalidate. As soon as you get the validation passes, which is what's displayed in this GIF, so you drag and drop in there, you get a pop-up, it'll say validation passes. At that point, you know that it's adherent and really compliant to the terminology that we're using for ReDU. And all you need to do is upload it to Massive then, which is probably the least intuitive bit because people don't often interact with this particular function in Massive too often. So Massive, again, this is the data repository. So we're adding the metadata to the data repository, and then we're going to automatically call the information into ReDU from there. So the way that this operates is it looks for any file in Massive with the name GNPS _ metadata.tsv. So you have to rename your file, whatever it is, to that exactly. If it's off, if it's capitalized a little bit, it will not be automatically pulled into ReDU. So this is an automation thing. So, but the way that you actually do it is, so you create the metadata, delete the extra rows, download it, validate it, and then you rename it, the GNPS _ metadata.tsv, and then you upload it to Massive. So if you go to Massive, so that's massive.ucsd.edu, and then you can find your Massive extension. Again, this is the number that you would put in column 1. I think there's a question about that. So that's a unique identifier to your data set. There's a button there that says add files. So if you click that, it gives you the ability to add more files in this right-hand side of the screen. The title here is simply for your record keeping. So you basically know what this is. So all the jobs in GNPS, like Massive, are essentially recorded and are there for you. The general description is something you can add. So, I commonly say, like, adding metadata to this thing, and maybe, you describe the study a little bit. There's a minimum, I think, of 50 characters that you have to add there. But most importantly, you want to add this file to the supplementary files location. So that's this bottom left button. And when you click that, it brings up this kind of pop-up window. And if you use GNPS, or if you've ever done molecular networking, this is the same kind of selection window. Oftentimes we operate on the first tab here, which is select file input. And that's how you would select files for, say, GNPS molecular networking here. And this is maybe a little bit hidden for people. There's an upload button as well, upload tab. So if you click on the upload tab, you can select the folder that you're operating here in, and simply drag and drop that file into this window here. There is a limit, but I think all of the metadata files are anything that you would contribute. We've tested on up to 3,000 files at a single time. So you can use this simple upload, drag and drop to do that. So when you do that, you can then select it in the supplementary files folder and then submit that job. You get a job link, it says complete, and essentially at that point, you're done. The ReDU, oh, sorry, there's a jet fighter. The ReDU system essentially it updates periodically, and it will go to Massive, find those metadata files, and try to pull the information into Massive, into the file selector. It won't immediately show up in the PCA plot, in the Emperor plot that I first showed. That is updated, that update cycle is a little bit longer. However, the file selector, it's pretty fast. Like I've seen it happen within a couple minutes, at the latest a couple hours, depending on the timing of when you actually contribute it. So I think that's it for how you actually add your data to ReDU. Unless there's any questions, I'd like to show people what it can do next. Because I think that's really what that should inspire you to actually start to utilize this system. Okay, any questions real quickly? Chat or audio, it's fine. Give it like a couple minutes here. Oh, there was one comment made by Ming and Julia. So the data does need to be public in Massive. So Massive data sets are not automatically public. So when you create it, you'll get a task completion. If you click on that task completion link, there's a button that says make public on Massive. So that's the way that you actually do that. And the way that you check whether or not your data is actually been input into ReDU is if after you submit it, again, wait at least a couple minutes, because it's not super fast because we're again looking for the entire repository. But you can go to ReDU, go to the file selector that I think Christy's going to show you right now, and there's a button that says Massive Dataset Extension. If you click on that, then it should list all the Massive IDs that are included in ReDU, and there you can actually search for your actual unique Massive extension number, as well as it displays how many files. So if you know that you have 100 files in that Massive ID, but you only see 50 included in ReDU, then something went wrong with the validation step. Either there was a term that somehow snuck through the validation, or we haven't updated, the system isn't done updating. So that could be why. What's the difference between, oh, okay, that's a great question from Matt. So the difference between the metadata that we are talking about in ReDU versus say like metadata that you would have just as you're doing your experiment, right? I think Matt is on the right path. It's essentially a formatting thing, right? A controlled set of terms so that we know that we can easily select things across data sets. So the simple answer is it's a template, it's formatted in a particular way, but otherwise it should be no different than the information that you're kind of capturing about your study anyway. It may capture things that you just kind of intuitively know, right? Like you intuitively know this is probably a study, for rats, but not everyone else will know that if you just deposit the data. So that's why we're asking some of these basic high-level questions about, really what the data, how it was collected, and kind of the technical things that go into it, like extraction method that may actually shift things around. So in many ways, you should be able to copy and paste. If you already have existing metadata and data in Massive, you could go back, and this is what we did within the lab, and retrospectively curate basically this ReDU metadata by copying and pasting and making sure that the terms are adherent. So again, you can literally just open up your existing spreadsheet of information that you may have about your samples and copy and paste urine. Now, you may call that human urine. We only allow the term urine, so there's maybe some, compatibility there that you need to flex, but that's essentially it. Okay, let's see. Can you use the ReDU template ahead of time? Yeah, and actually that's the recommendation now. If you go to GNPS and the documentation, we recommend that you use the ReDU metadata template as your starting template. And again, when you make a personal copy, that is your copy. You can do whatever you want with it. So you can add how many other columns that you want, and you can upload all of that to Massive. It's not automatically pulled into ReDU. Only the columns that are in that base template essentially are called into ReDU. But you can use that for your own purposes as well, and basically have a unified way of collecting the data. So that's the new recommendation for using GNPS. There's also instructions on how to convert this template into something that's compatible with molecular networking. So essentially, you just need to add attribute to the columns that you want to see in the molecular networking job, and that way you also have a consistent way of displaying that information. Let's see. Then open the template. Does it have a place for referencing published data? So it doesn't have a place for referencing published data. Hopefully the data is in a public repository. So currently we're only using Massive, but there's some talk about also like pulling in data from the other databases like Metabolites and Metabolomics Workbench and others. And the second point is, does it generate a DOI? And this could be the last question. The DOI has, oh, perfect, Ming just answered it. The Massive data set has a DOI that you can reference. Okay, I want to make sure that we have plenty of time. Again, all of our contact information is on the last slide of this set. So email and Twitter are great ways to reach out to us, as well as the GNPS form. Okay, I think, Chrissy, do you want to take over?
I am ready to take over.
Okay, cool. I'm gonna stop sharing. It's all you.
Okay. Hi, I'm Chrissy, and I'm just going to be telling you a little bit today about what you can actually do through the ReDU website. So there's a few different cool features that will allow you to just kind of like on the spot explore some of the data or maybe analyze your own. So if you look at the ReDU homepage, there's two sides to it. There's the analyze your data and analyze the public data side. So I'm just going to talk a little bit about what you can do with your data if you've run it through GNPS. So one of the things you can do is you can compare your data to the public data via multivariate analysis. So if you were to click on this screen or that link, what happens is you see this. At that, oh, whoops. At that point, you would then have the ability to enter your GNPS library search task ID. So something that Alan had kind of touched on is basically this integration between ReDU and GNPS. So if you've uploaded your data to Massive or you're just using GNPS for like library search, what happens is it'll generate a task ID. So you can find that like on your GNPS homepage. Or it's also in the URL if you're running a GNPS job. And then you can input that task ID, and if you click Create PCA, it'll then allow you to pretty much project your data into multivariate space. So yeah, so you can find it on the Jobs tab at GNPS or in the URL. Or you can just click for an example. So if you want to quickly click, it'll then display this kind of three-dimensional principal component analysis, which is really cool. So it's visualized via Emperor and it's three-dimensional. I don't think the slide quite does it justice because you can like zoom in and zoom out and then spin the things too, so you can like go to different portions of the data. As well as if you look at the right side of this screen, you will have the ability to go through the different metadata columns and then color all of the samples accordingly. So that's where the curated metadata starts to like really come in handy. Is, for example, if we've labeled your samples rat and you thought that was very obvious, well, now we have 40,000 roughly samples of public data, and what you can do is you can go through and color all of the public data that's in ReDU according to what species it comes from. So you'll see, like, for example, like a cluster of blue points, which you know corresponds to all of the rat samples. So the way this is created is basically all of this is using only annotated data. So each of these points represents a single sample that is within the ReDU database. And every sample is basically plotted according to what is present or absent within the data. Oh, okay. So
there was a question in the chat before you move on.
Oh, I can't see the chat right now.
Is it doing and the question was, is it doing any scaling, centering, normalizing of the data?
Yeah, so all of the data would be like mean centered when you're actually like going through the calculations of it all. But it's PCA, so it's basically just Euclidean distance.
And then what we're actually looking at are basically the presence or absence, basically the observation of an annotation within the data set. So, you know, for example, if you take one chemical like caffeine, essentially it's binary, seen or not seen, based on library match within a sample.
Yeah. Alan, is this an animation?
Oh, that would be Mauricio's. Yeah, I think.
Yeah, nice. So this is just like a short little animation which shows you like what you can actually do within the interface. So yeah, that's just spinning it around, and then it looks like he would be going through the different tabs. Oh yeah, so in Cytoscape, or sorry, not Cytoscape, in Emperor, something you can do is you can, I think he's doing scaling right now, which is where you can like basically change the sizes of the points or so he'd colored a specific subset of points and then wanted to make them larger so he could be able to actually visualize them against the background of 40,000 points. And then making other ones transparent. So it's really cool because it'll allow you to like pick out exactly what you want from a plot, which would normally be pretty overwhelming, I think. Okay, cool. Okay, I'm just gonna pause that. Does anyone have any questions?
And just to add, if I could, you know, this is really meant to be kind of high level context, you know, assessment of your data. So if you're running blood from humans, you can kind of see in general where that data falls in the context of all other blood that's been run, to either say, you know, as a QC measure, saying, you know, it kind of fits the chemistry that you expect, or if you're operating on a very different system, you know, a lot of natural products chemists, you know, that you're interested in the unique chemical space, right? So you may want to observe your samples, you know, very distantly from everyone else's. So those are kind of, you know, this is really meant to add context to your data.
Yeah, that's totally true. Okay. So another section, if you then navigate back to the homepage, is you can also co-analyze your data at GNPS. So now we're going to get into a little bit of the file selector tool. And whereas the first portion of this using multivariate analysis allowed you to contextualize your data through PCA, what this is going to do is it's going to give you a whole bunch of different options that you can use to then launch analyses, as well as parse data down so that you can not only contextualize it in the span of like 40,000 samples, but if you want to sub-select samples, it'll give you that ability. So this is what the file selector page looks like. And if you just look at the page on the home screen, there's three main parts to it really. So there's the selection summary, which will allow you to keep track of what samples you put into what group. There's the actual file selection portion in the center, the yellow buttons, which each of those buttons represents a column in the ReDU metadata. So if you want to think of it as an Excel sheet, it's just like filtering an Excel sheet. So it'll let you parse apart the data. And then on the far right is the attribute filters, which will again allow you to keep track of what data you've filtered in what way. So if you scroll down to the bottom of the page, for example, if you see this is pointed to NCBI taxonomy. So if you were to click that and then scroll to the bottom of the page, it will show you all the different NCBI taxonomies represented within all of the ReDU data. So for example, you have Homo sapien data and you have all these different like types of species. And yeah, so it'll be the NCBI number and then the actual written word of what it is. And then it'll allow you to either filter or add it to a group. So if you hit the filter button, what it will do is it'll then allow you to apply a secondary filter. So let's say I wanted all human samples, but I wanted a very specific sample type. Like I wanted to look at all human urine samples as opposed to whatever sample types there are in present. Clicking the filter button would then allow me to take that and refilter it again. So it'll also display the number of files. So it'll tell you like that's the total number of human files within the data set. Alternatively, you could just take all of the human samples and start adding them to these groups, G1, G2, G3, G4, if that's something you would want to do. So then as soon as we apply this filter of looking at all human samples, if you scroll back up the page, it'll show you to help you keep track of like what filters you've been applying. And then, okay, so if we're giving a second one, it'll allow you to select like a different filter. So, okay, and again, now body part. So let's say, yeah, now we have it filtered by human and we want to look at human blood plasma. You can now add it to different groups. So it'll still, again, display the number of files as 678, and then we add it to the first group. So for the demo, we're going to add blood plasma to group 1 and then urine to group 2.
We had a question whether the person or lab who submitted the data can be a filtering category.
That is not a filtering category, unfortunately.
Yeah, some of that information is captured already in massive when you contribute the initial data. That is something that we've discussed about including. So if you think that's useful, that's great feedback. The best way to kind of add those suggestions is through our GitHub page and just make an issue real quick. That way, you know, there's a record of, you know, what everyone kind of is requesting. But I think something like that, at least the contributor, I think we can capture some of that information and possibly get that.
Well, we can definitely capture it because we're capturing it. I think also once you do filtering, when you do analysis, then you should be able to filter for lab and a person that uploaded. But we do not have a direct filter in ReDU.
Yeah.
But it
yeah.
might be a good idea to add.
Yeah, that's true. Another thing is if you were looking for like specifically your own data and you know what massive IDs you had, you could always go to like the data set accession and then you could try and like pick out all your own massive IDs if that was something. So that's currently something that's supported if you needed that. Right, so then after you add different files to different groups, it'll show you on the selection summary as well the number of files that end up in the groups. Again, to help you keep track of like what's where. So at the very bottom, once you've fully like filtered all your samples out and you've added them to the different groups, you have a number of options for like things that you can do. So for example, you can set up some co-analysis at GNPS, molecular networking. So basically what all of these different options do is they allow you to like link out to other services either within our own website or through GNPS that allow, and then you can basically just perform that analysis on the subset of the data that you've now selected. But you have to have a GNPS account and you need to be signed in before you click the button. Otherwise, you're going to have to go back and like reselect everything and take care of that. Yeah. Alan, is this where you're going to take over again? Okay.
I think, yeah, I can, if you want me to.
Oh, I don't remember what slide.
I think we're still going to
Sorry.
cover this one, but.
Okay, I can still cover this one. It's okay. So for example, if you were to link out to GNPS molecular networking, this would just be what the homepage looks like. And what you can do is if you look down to the file selection option, you would then be able to see like all those files you selected into group 1 and group 2 would again be in group 1 and group 2. And again, the number of files should match what you've selected from the file count in the previous page. And then this is a good point at which you always have the option of adding your own data. So if you wanted to then like, you're like, oh, I have another 100 files I want to co-analyze, you can just add them into a separate group and it would work fine for the same group, I guess.
And just to kind of link back to what I initially kind of said in the beginning, you know, this is essentially the shortcut to find those files that are most relevant to you in the database. So, you know, if you actually look at all the different massive extensions and file names included in that G1, you know, that's blood, you know, it ranges from like, it could be 20 or 30 different data sets that you're pulling data from and collating it and pre-populating essentially this information needed to do molecular networking. And it's really, really informative to start doing this with your own data, as I'll kind of mention later. But essentially, if you have blood data, by adding other people's blood data, you can better understand some of the annotations in your molecular network. Sorry, Chrissy, all you.
Yeah. Yeah, well, the cool thing is too is if you didn't have this kind of like systematic metadata and you needed more samples to do that kind of analysis, it would be extremely hard to go through massive and pick it out all on your own. So what the file selector is really useful for is just efficiency in adding data for additional analysis or comparison in a really efficient manner. So another option after you've filtered out your samples is you can just launch a PCA of the selected files. So this is a little bit different because while as before we were exploring like the chemical space of all 40K, however many samples, this will build you a specific PCA for whatever you've sub-selected. So if you've sub-selected human, what was it, urine and blood plasma, then it'll just calculate a brand new PCA for you and you will be within the same Emperor interface and whatnot. So you'll have all the same options for coloring points by other metadata categories, making them smaller, larger, and basically just being able to play with the data. So if we then go and we color these human samples that we've sub-selected by, I think this is comorbidity, now we can be able to see that all the samples in humans for urine and plasma will cluster whether they've got diseases or no diseases reported. So that's really cool. And that's kind of what we would hope to expect as well. So it's kind of a good sanity check. And then it'll show you the legend, of course, on the right. So anytime you are coloring by metadata category, you always have the option to change the sample color and it'll always tell you like what corresponds to what. So then you can also color them by ionization mode, for example. And as you can see, like the negative mode clusters together and separately from the positive mode, which is good because if it didn't, that would be bad. And you can just do a lot of different options like this. So anything that's captured within the ReDU metadata, you can pretty much sort by it or color by it within this plot system. Okay. I think
it's back to me. Okay.
Alan, you're muted.
Good call. You know, that PCA example is kind of a pet example. Obviously, you know, blood and urine should probably separate based on the chemistry that you observe in the data. But those are kind of examples of some of the technical things that you can search through. You know, it's like, okay, well, why are things separating? It could be extraction method, it could be the instrument, it could be, you know, ionization mode because you observe different chemicals there. What we found out is, in general, say blood tends to look like blood, regardless of, you know, the different extraction method or instrument or year that is run, say compared to something, you know, like your skin or feces, which is a good sanity check for, you know, metabolomics in general, in that, you know, we're at least capturing consistently some information that's characteristic of these different sample types. And so, okay. Oops. Can you see my screen? Okay. So I think those are really useful tools. You know, be able to project your data, get the extra context by using the PCA plot, as well as you know, being able to contribute your own data and really co-analyze it using molecular networking. We kind of jumped over the molecular networking and kind of what it is and specifically, but there's a couple of videos that, from a couple of weeks ago based on for that. That we posted on the workshop site, as well as there's just information in general in the documentation, a lot of information on molecular networking. So I want to talk a couple minutes about some of the unique tools that we built now that we have this kind of repository information. And one is Chemical Explorer. So that's found on the right-hand side of the home page. And really what this is it's a, oh, okay. Doesn't want to behave. Must be an animation. Sorry. Oh, that's even worse. I apologize. Okay. The Chemical Explorer is essentially a way of, if you're interested in a particular compound, you can use this to determine where it's been previously found in the repository data. So again, Chemical Explorer is here, and the resulting page is something like this. So this is a huge amount of information. We tabulate all the different annotations that have been found through spectral library matching. So this is, you know, from the GNPS library, which also integrates other libraries of MS/MS spectra. And essentially it goes through, we do a giant library search on the data, and we report the pre-calculated, pre-computed values for you. So there's download buttons on the left-hand side. So if you want to download all this information, we give that to you, as well as a search box. Control F also works if you're using PCs to search for the compound name. So in this case, I searched for citalopram, and we see there's multiple hits. So these correspond to different MS/MS spectra matching different MS/MS spectra in different files matching different references. So that could be different collision energies, different instruments, different, you know, set of conditions that maybe the MS/MS spectra are slightly different. In this case, I selected the line with 82 files. If you click the View Files button, then it tells you explicitly all the files that that MS/MS was matched in. But really, what the most useful feature, I think, is the View Annotations button. So if you click the View Annotations button, it brings you to the subsequent page And here we're basically doing some basic tabulation of information here. So we're looking at the files in which that MSMS was observed and matched. What is the metadata essentially associated with that match? So in this case, if you look at citalopram, you can start to, we have all the basic information tabulated here, but we also built in a graphing tool. So if you go to each one of these tabs, say, I think this is disease, reported disease, you know, the most often time that citalopram is found is in people who report being unhealthy, and NOS is not otherwise specified, right? So it's just generally people who may have disease for unknown reason and a chronic illness. So as this is a medication that may make sense, you know, we don't really have the fidelity to ever explicitly say, you know, why, you know, because that would require an incredible amount of metadata capture. And I know you don't want to spend a month, you know, just filling out a spreadsheet template. So essentially you can go through, you can look at the year, the different instruments that was collected on, as well as the distribution within samples. So Peter actually posed, I think, a really good hypothetical question, which is, you know, how do you really know what, you know, chemicals are distributed in, say, kids under, you know, age 2 versus, say, an adult population? Here you can actually, instead of saying that high level view, you can go more explicitly and say, okay, well, how does chemical A distribute across age? And that actually, so if you look at, say, the bile acids, you can see a difference there with age, and you can start to dive in some of these age trends. And hopefully as this database grows, you know, it becomes ever more accurate as people add the variety, all the different variances that could contribute to this data. So
I think age is one category. I think for people who care about natural products, you may be able to ask the question, okay, what country, what longitude, latitude, et cetera, et cetera.
Yep. And the organism through the NCBI information seems to be very useful and informative. And at least for me, you know, in my own research, you know, oftentimes I'm asked, you know, I'm interested in this molecule. What type of sample should I collect from, you know, human subjects? Blood, urine, saliva, you know, what, and at least here you can go through, you can search for that chemical and you can empirically say, okay, well, you know, it's most often observed in the repository in, say, urine samples. So maybe that's the way that you want to start to capture some of that chemistry.
Yeah, another example would be really interesting, for example, where people in the microbiome field often want to know, is this made by a microbe or potentially by the host? And one way to sort this out is, let's say you search a certain spectra or a specific molecule, and you find out that it's in a lot of cultured microbial data sets. That is one way to build a hypothesis that it's probably of microbial origin. And so these are the kind of ways that you can use this kind of information.
Yep. So there's a lot of potential in here. And again, if you can think of interesting things, please make suggestions on GitHub or email us. So again, this is just a zoom in, kind of what I was talking about. So in this case, citalopram, looking at what the sample type is observed in. So in this case, it was plasma. Obviously, these values kind of shift depending on, you know, how many files are contributed. So, you know, again, more data in here, the more accurate these should become eventually, as well as some health category. So this is kind of a pet example, again, for you to kind of understand some of the things that you can do. And then the last bit is the reanalysis of public data. So this is, it actually links to the same file selector that Chrissy actually covered. The orientation here is slightly different, and we have a couple different things. So I want to show you one example of what you can do with molecular networking at this scale. So this is selecting human blood, urine, and feces. So that, at least at the time it was done, which is a while ago, it was over 6,000 samples. And while we have a limitation on doing molecular networking, a suggested limit is 2,500 samples. If you select more than that, you could run into memory issues, in which case you have to contact us for more resources. And Ming's kind of the ultimate judge of how those resources are distributed. But this is an incredible amount of data, right? And we're networking on over 5 million MS spectra in terms of humans, right? And that leads us to 88,000 essentially of these nodes, right? Which are these circles that are pinpoints on this slide, which each one of those we assume are a proxy for a real chemical, right? And you know, the orange bits are essentially things that we can give a name to. So there's a lot of unknown space, but, and this is the argument that I would make for doing this, or even including your own data in this type of analysis. By doing so, you can further your annotation. So in GNPS, one of the unique things that we allow is anyone can add a reference spectrum based on your own belief that this is something that that you observe, you know, based on your interpretation of things. And molecular networking gives you the ability to work from analogy, which is a huge way of reducing the possibilities in terms of what an unknown could be, right? So in this case, the shape of the node corresponds to whether or not it was annotated or not. So the diamonds were the only things initially annotated here. So this is clindamycin. So it hit on reference spectra to clindamycin. All the other ones were unknowns. Again, this is, you know, the compilation of 6,000 samples. And what you can observe is this ranges from 1 to 6 data sets. So some of these most commonly observed, we'll call them analogs, metabolites, they were found in a lot of data sets. But if you look at some of the connections, they wouldn't inherently be made. So for example, between this 425, which is the clindamycin, to 411, these were never observed in the same data set. So you don't ever get that direct linkage if you're just operating on your own data. So going through basically, you know, interpreting the manual differences between the masses and the MS spectra, you can start to assign all of these unknowns and look at their distribution in all the different bodily fluids. So in this case, it's really supposed to intended to show that, you know, by combining this public data with your own data here, which I actually added my own data here, you know, I get a lot more information that I can start annotating things and start to make more sense of my own data. Let's see.
Alan, before we go on, could we take one step back to the Chemical Explorer and show how we can download the outputs?
Yes, definitely. Okay, so the Chemical Explorer, so this is the first page that you hit. The download here will download just the list of all the annotations found in the number of files. So that's partially useful. If you click on the View Annotations button, then there's a download button underneath the plotting tool. So there you can download as CSV, Excel, or copy it to the copy and paste it, say, into like whatever spreadsheet program that you want. So that's how you would download those as well. If that makes sense.
Does that cover the question?
What is the best way to download information about the samples? But can you get submitted data? Oh, okay. So maybe this is a slightly different question. So this is really pertaining only to this particular compound. If you're wanting to download just all of the information, you can do that on the home page. There's the download database table and download annotations, I think is the link. There you can download all the metadata, or you can download all the annotation information, which is really where Chemical Explorer it kind of operates on that table in a way and kind of simplifies it. But there you can download everything. So if you're interested in, I think the question was, oh, okay. Anyway, you can use that table to do all kinds of operations. So the question is, if I find a compound, can I use the link here to find what samples that compound is in? So the answer, the answer is yes. I'll let them answer in the chat. But briefly, if you go here, you can see the view files button gives you a list of all those files. Get to it. Okay. So large-scale molecular networking, it's useful. You should try it. The other function, real quick, because I want to leave plenty of time for questions. The group comparator. So this is something unique to ReDU as well. So if we go back to the example that kind of Chrissy talked about, which is if you just select blood into group 1 and urine into group 2 as kind of a pet example using the filtering, we can start to then ask using group comparator, how are these two groups different based on their chemistry? So if you select your groups, you click group comparator, and again, we allow up to 6 groups for comparison. The first column are all the annotations. The second column is the number of files in which the annotation was found. And then we also provide a normalized percentage so it's a little bit more comparable in case you have class imbalance between group 1, group 2. So for example, you can see this indole 3 lactic acid hit. It was observed in 84% of the blood plasma samples, but only 22% of the urine samples. So here you can start to say, okay, well, how are certain particular chemicals different between groups that you may select? Right now, this only operates on the public data. So the way that you look at your data is you could add your data, select all the files in your massive extension using that filter essentially into group 1, and then add all other files that you're related to maybe your particular question in G2. So if this is somewhat like Peter mentioned, a lot of natural products things, if you're looking at bacterial cultures, you could add your data to G1, select all other bacterial cultures in G2, and start to say, okay, well, what is unique to my culture system as opposed to whatever is in the repository, as a way of really comparing and contrasting. Or if you have specific questions in mind, there's a search box as well. And this is all downloadable. And on our GitHub, we have an R code. There's code in R as well as a notebook, a Python notebook, a Jupyter notebook that you can use to plot some of these using bar graphs, for example. And then the last thing is this. We mentioned Chemical Explorer just a while ago. Here we give the ability to do Chemical Explorer and generate that information based on the metadata on a specific set of files that you select. In this case, we're only operating on things selected into G1. So make sure that everything, if you want to add multiple things, are all included in the G1 group. Otherwise, they'll be ignored. And essentially, you'll get the same kind of information as we display with the citalopram. You get the tabulated list of annotations. If you click on the associations are now calculated from the subset of files. And if you see all zeros, it means that it wasn't observed in that system. Is how it's currently displayed. So if there's no other questions, this is open time for questions about template analyses that you can do, any of the other functions. Otherwise, thank you all for attending. I know it's been a little bit rough. My internet was kind of slow, but I hope you really see the value in this, and we're welcome to any suggestions that you might have. So thank you.